Log on, tune in, find out. Another good idea from Cambridge.
Next speaker is Jan from the University of Leuven. So just pass it to you.
So thank you very much. Let me first of all thank the organizers of this workshop. It's a pleasure to be here. And a few warnings. I am an engineer. I'm not a statistician. And I will be talking about applications. So let's see what happens. Now the title of the talk is Optimal Experimental Design for Nonlinear Systems, Application to Microbial Kinetics Identification. Now this is joint work. We have been working in the team with a number of people, and these people are still with me as postdocs in biotech. Biotech stands for Chemical and Biochemical Process Technology and Control, one of the 3 research divisions in the Chemical Engineering Department of Leuven University. Now this is an overview, and you will find it later in the slides. It's basically a set of 5 websites, and I start with a few words on our Center of Excellence, Optimization in Engineering. Then I continue with the major part of the talk, what we do in our research division in biotech. And then this is just to conclude what our platforms, where we try to transfer the things that we are doing in the department to industry in many different areas. So let's start a little bit on that Center of Excellence Optimization in Engineering. These Centers of Excellence, these are kind of virtual institutes funded by the university for a period of 7 years. And for this second phase, we are already in the second phase, the period 2010-2017, 5 departments, as you see them listed here, participate in that Center of Excellence. And we deal with quite a number of different problems and challenges, and I go immediately to the grand challenges, and you will recognize many of these being relevant for optimal experimental design as well. Robustness against uncertainty, nonlinearities, large-scale problems and distributed computing, fast and reliable algorithms, and real-world applications. Now the Center of Excellence, as I explained, is composed of 5 departments, and these work together in 6 working groups on a fundament of optimization in education and training in our master and graduate program. Dynamic and embedded optimization, data-driven modeling, black box modeling, parameter and state estimation, this is basically the issue of today. Shape and topology optimization, advanced linear systems, analysis and control, and PDE constraint optimization. On top of this, we try to integrate all of this in real application projects. And as you can see, optimal experimental design is all around over the place in all of these issues, in all of these themes that we try to cover. We do no longer work as independent departments. We collaborate over the traditional borders of the departments to work on concrete problems, people from chemical engineering, electrical engineering, software engineering, and so on. My own research division, biotech, this is our research matrix, and our mission statement is model-based design, optimization, and control of chemical and biochemical conversion processes. And if you look along the rows of this matrix, we work on system theory, modeling and identification, monitoring, optimization and control. And of course, optimal experimental design is in the first row of the matrix, where we apply this to chemical and biochemical reactions and reactors. Microbial dynamics also occur in biological wastewater treatment systems. It also occurs in predictive microbiology with respect to food safety, food spoilage. Obviously aware of the number of people that die every year because of food spoilage and food poisoning. It's a dramatic number. In the United States, the last number available is comparable to 1 typical Belgian city a year. So that's a huge number. Also, the outbreak of the EHEC bacteria and E. coli bacteria demonstrates also the relevance of this type of research with respect to public health. So far the introduction, so let's now come to the major part of the talk, optimal experimental design for microbial kinetics, and I will discuss some problems. What can we do with optimal experimental design? And as a first very long case study, we concentrate on predictive microbiology, an easy problem 2-parameter problem, a 4-parameter problem, and then a second case study on bioreactor identification, where we discuss a bit or introduce a bit trade-offs in optimal experimental design and trade-offs in design and process control. And you stopped me when you got enough. I prepared some slides. I have to say also that, well, this might look easy. I picked easy problems just for the sake of trying to be as clear as possible for this presentation. At this moment, you are obviously tackling more complicated problems than that. Secondly, all experiments that you see are being done in the lab itself. So we are a lab that is concentrating both on the experimental part, doing the real experiments with microorganisms, and then trying to identify appropriate models. Parameter estimation, of course, one way to do this is by minimizing the error functional, as you see it here in continuous time or discrete time. And typical problems when we are dealing with bioprocess models, no unique estimates, highly correlated estimates, the two are also correlated, low accuracies, convergence problems, it's biting your own tail, optimal experimental design. You have to start from some nominal values, but these appear in the design itself, so it needs to converge, the scheme that you set up, in an iterative way. Estimation problems, they arise because of, it can happen that there are small sensitivities of outputs, certain outputs with respect to certain parameters. In biological systems, this is most often the case. Only a few parameters are dominant. Model parameter correlation, sensitivity to measurement noise, complete lack of measurements, low frequency of measurements. If you compare to a mechanical or an electrical system, if you have a data record of one experiment composed of 12 data points, we are quite happy. That's not big. Model structure limitations and so on. So if the data are not sufficiently rich, then of course there is no unique parameter estimate possible. And there was a question this morning, I don't remember by who. These parameters, P1 and P2, they're highly correlated. Any combination will model the data. But of course, if you're after the true, what the hell is this? Do I want to proceed? I should say yes. No? No? Ooh. Thanks.
Otherwise something will happen.
Well, we'll see. At least it's not optimal. But thanks for otherwise. So these parameters are correlated definitely. If you're looking, say, for a maximum specific growth rate, you cannot find it from an experiment leading to this identification functional. What we need to do by performing a better experiment, an optimal experiment, is to reshape that functional and hopefully end up with a unique minimum. And also the previous speaker gave an excellent introduction on all problems and pitfalls that can arise, because this is all local, it's not global. Experimental design for parameter estimation, we assume that we have a correct model structure, that we have time invariant model parameters, and then we hope that we can find a unique set of parameters P star that characterize the system's dynamics. So it's basically a mathematical methodology to design optimal dynamic input profiles, maximization of information content in experimental data. We also can concentrate on selection of informative experimental zones, parts, windows in the data. I will also give an example of this. With the idea to end up with reliable and accurate parameter estimates, the parameter vector P, by using a limited, hopefully limited set of static and or dynamic experiments. Now this issue is extremely important in the area of biology, because these experiments are extremely expensive, most often. Identification cost, well, I will not repeat the basic theory, but by some linearization and first order approximation, you immediately, you can see it almost from a distance visually, happening that by linearization and expanding and keeping first order terms, you end up with a Fisher information matrix. And here it is, it's content quantification. Q is the weighting matrix. There is the matrix of model output sensitivities. And here is our dynamic optimization problem. T is what I call a certain input to the process. and I am after the dynamic input. It could be a dynamic input as function of time t that optimizes a scalar function of that Fisher information matrix, and that's my optimal input, that's my optimal design that I will then apply in practice. Criteria, it has been discussed also this morning. I have a better plot on this, but also that one was already shown this morning, so I will not repeat it. A criterion, D criterion, E criterion, many different and also new criteria are being developed. In one dimension, obviously, the idea is that we, as I said, we reshape the identification functional. So by reshaping, this one definitely has a better, a smaller confidence interval if we are after a smaller confidence interval. Also, that is not to be taken for granted. It depends on what is the aim of your optimization problem. Especially when working with biological data, you should keep in mind that the quality of the experimental data is not that high. So you should keep in mind that an absolute accuracy is maybe not the best to aim for. So as a first rather long case study, optimal experimental design in the area of predictive microbiology, typical microbial dynamics. It can be growth, survival, inactivation of microorganisms, and we consider them in changing environmental conditions as these occur in food products, in reactors, in biofilms, in many different, on our skin, in our system itself. And they are influenced by environmental conditions, dynamically changing temperature, pH, water activity, and so on. Typical example that we study in the lab is food quality and food safety. And of course, the specific use of this type of models is in the food industry, government, storage guidelines, food handling by regulatory bodies, legislation, and then last but not least, risk assessment. A huge area of interest these days. Microbial food safety and quality. So what we want, what we mean by this is a reduction and or prevention of microbial growth, definitely of the pathogenic organisms, like E. coli, like Salmonella, like Listeria. Microbial dynamics, they are determined by environmental factors, temperature, pH, and so on. So here you see a typical from farm to fork situation where we have here the ingredients being processed, transportation and storage, and finally consumption at our homes. And this is a typical simulated, it's a simulated temperature profile, but it's more or less realistic. Here there is some abuse of temperature during the processing, maybe, which can give rise in blue to an increase of microorganisms. What is the most critical part, and we all know it, is to keep the organisms under control during this very long period of time after processing, during the transportation and storage up to consumption. And for this we need as accurate models as possible. General expression, and it will come back a number of times, the NDT is a specific rate times n. That's a basic equation for microbial evolution. And I did not invent it myself, you can find it in any standard undergrad textbook. So if mu is negative, we have inactivation of the organism as a function of time. If mu is positive, obviously, then we have growth of the organism as a function of time. Now these are the data that we typically work with. This is the natural logarithm of colony forming units as a function of time for a specific sort of E. coli K12. And this is the simulation with a mathematical model. And we can distinguish three phases: the lag phase, what we call the exponential phase, and the stationary phase. And the exponential phase is of course easy to describe. On a natural log scale, it's a straight line. The more difficult parts are the lag phase, and it's indicated here how we compute it. So this is the lag phase as we compute it, intersection with the tangent line. Then we have the exponential phase with its maximum specific growth rate. This is the key parameter that we want to know, maximum specific growth rates. And finally, the stationary phase, when the cells decide to stop growing because there are too many or whatever reason. At constant temperature, we still have our basic expression, the ndt is equal mu times n, but you can easily see that mu is no longer a constant as a function of time. Here it is a constant, here it is not, and here it is not. So therefore we have the model of Baragni and Roberts, where this is still my global mu, and this factor, which is also increasing exponentially, converging to 1, is responsible for this adaptation phase. This mu max factor is responsible for this phase, and this factor, the logistic factor basically, is responsible for reaching the asymptotic phase at the end of the curve. So you see it here, lag phase, exponential phase, and stationary phase. Now for a two-parameter problem, I will give you some more or try to give you some more insight. And it's work that we published. The first publication stems already from the years, about 10 years ago, but we are still continuing in this area on multi-parameter systems. What you see in this plot are experimental data collected at different but constant temperatures. So this is a growth curve at 16 degrees C, one at 23.1 degrees C, one at 32 degrees C. And what you see is that the slope is different, of course, with respect to the temperature that we apply. For each experiment, the temperature is constant. If we compute the slope of this model fit and we plot it against temperature, then you typically get a picture like this. So this is the slope, the maximum specific growth rate of the growth curve as a function of a temperature. And it typically behaves like this. Knowledge of this curve is essential because it's a characteristic of each individual microorganism in a specific environment. In the first stage, we are interested in the suboptimal region, the suboptimal range, which is the range from the minimum temperature for growth up to the maximum temperature for growth. And as you can see, if you take the square root of the maximum specific growth rate, then this maximum specific growth rate, the square root of it, almost behaves like a linear relation with respect to temperature. So that's why, and here you see experimental data that we collected in the lab. Keep in mind that each of these dots represents a full growth curve. Okay? And this is a specific growth rate corresponding to one of these growth curves. Okay? So you see here about 20, 25 growth curves. This is the model. The square root of mu max is a parameter b, t minus t minimum, b being the slope, and t minimum obviously the intercept with the x-axis as you see it plotted here, and this is b, the slope. Okay, if we now combine the growth model with the square root model, then we have parameterized our model as a function of time and temperature. The mu is now explicitly function of temperature as we see it here, and we can even allow temperature to be function of time. And we are looking for the parameters b and t minimum for that specific microorganism. Okay, that's my standard problem for a few minutes. And of course we would like to apply the model then later on under time varying conditions, because these are the conditions that will occur in practice. Now, as you have seen, this is an example. So this is a typical temperature profile that could occur in practice, and this is a prediction by the mathematical model. You might say this looks completely bullshit, but I will illustrate with real data that is very realistic. Most importantly in biological systems is that apart from the prediction of the output variable, whatever it is, we are also interested in the uncertainty on that prediction. And computation of that is of very huge importance, for instance, with respect to risk analysis. So we are trying to identify the parameters b and t minimum. Let's first sit at the computer and then go to the lab. Fisher information matrix. In our case, we have one output variable, n, the population density, and two parameters, b and t minimum. This is our measurement noise, well, 1 over the variance of the measurement noise. which is rather big. You have seen the quality of the data before. Nominal model parameters, these are, we need to provide the iteration loop and initial guess, a B0 and a T minimum 0 initial guess of the two parameters. And we will be using the modified E criterion, and I know that there's many criticism against the E criterion. For people doing research, it has a certain elegance in the sense that, optimally speaking, the minimum value is 1. So just for computational purposes, it has some nice advantages. Later on, we will do exactly the same with the D criterion. So here is again our problem. Find a profile T as a function of time T in order to identify in a unique way the parameters B and T minimum with as output variable the population density N. This is basically our system. Relatively easy. Optimal design, so of the dynamic temperature input. There is a very important remark here. It says that the model structure implicitly assumes that the growth rate mu, all of this is the growth rate, as you remember, the N over dt is mu times N, that the growth rate immediately follows any change in the temperature. If we change temperature, then the growth rate immediately changes. You can already feel that an organism will not do that in practice, and we will see the consequences of this later on. As I said before, since the optimal design depends on our initial choices, we will perform a sequential experiment design also. That will be illustrated in a few seconds. So now we go to the lab. Here is our bioreactor, 5-liter bioreactor, 6-liter bioreactor, 5-liter or 4.5 working volume, all computer controlled. What do we measure? Temperature, dissolved oxygen, pH, rotation speed of the agitator, and so on. We work with an E. coli organism. Offline measurements, this is our output, the N, the cell density. We do it by plate counting. You could use another technique as well. So basically the result is colony forming units. Design of the optimal temperature input. Well, we said let's keep it simple. Let's start from a linear temperature profile, could be increasing or decreasing. So the degrees of freedom are the intercept and the slope. Okay? And at the same time, we try to optimize a quadratic profile, but we impose a lower temperature and a higher temperature for growth. Why? Because we want to stay within the suboptimal range of growth. We want only to identify the suboptimal part of the specific growth rate as function of temperature. Okay? So that's why we impose this T low and this T high. This T high is lower, a bit lower than the optimal temperature for growth. This T low is a bit high, a bit smaller, excuse me. This one is a bit larger than the minimum temperature for growth. This is also a second reason we cannot do any experiment at T minimum itself, simply because the experiment will last forever. There is no growth at T minimum. So the sensitivity there is also very interesting to look at. So these are our admissible profiles. Let's see what comes out, at least in computation. This is a linearly increasing temperature. This is the corresponding simulated output of the model. And this is our corresponding contour plot with the condition number. Not bad, not good either. And we limited the profile in between 15 and 35 degrees C. If we compare to an optimized quadratic profile, and it looks like this. We start at the lower limit, we go to the upper limit, and here it almost looks straight up to the upper limit. Condition number has decreased drastically. You see it also at the quality of the contour plots. This is much better than what we have here. It's on a relative scale, so you can appreciate this is about 25% of the nominal value that I show here in this plot. Now, if we look a bit in more detail to this profile, because you could say, well, where the hell is this quadratic profile? Well, it's mainly because the quadratic profile that we ended up with is dominated by this lower limit, so this part is not taken into account, and then it's so rapidly increasing, this is a scale 10 to the power 4, that we immediately reach the upper limit of 35 degrees C. Okay? So that makes that it looks like a step change, and if we then optimize a step profile, which is of course the most easy thing to do in practice, to do an experiment, instead of a quadratic changing temperature, we do a step change of temperature, so we shift from one temperature environment to the other one. That's very easy to perform in practice, theoretically speaking. Then we get optimal values like the lower limit is, or the T1 is immediately coinciding with the lower limit. The second temperature coincides with a higher limit, so we try to span as much as possible what we can, what we allow to span, and the degree of freedom is the shift from lower temperature to the higher temperature. And you see that from this so-called quadratic profile to the real step profile, the difference is negligible. Okay, so this is basically a step profile. There's a reason why we continue working with step profiles for the rest of this presentation. So the optimal temperature profile is a step temperature input that starts at the lower admissible temperature and that ends at the higher admissible temperature, 1 degree of freedom left where to shift as function of time. Pretty easy. Experimental validation. Experimental data at the temperature of 15 degrees C. Experimental data here continued at a temperature equal to 35 degrees C. Now, at first sight, we might say fantastic, or at least I might say that, or I did say that. At second sight, it's not that fantastic. What happens is the following. The model prediction does the following. Here we have the model prediction after identification at 15 degrees C, and this is here our model prediction at 35 degrees C. You might say, well, this is not too bad, but there is an intermediate delay induced because of this large 20 degrees jump in temperature. Okay, and this small delay is responsible for this short, relatively small delay that you see here as a function of time, but it's continued up to when we reach the stationary phase. Okay, that's important because we are not interested in the horizontal line, we are interested in the instantaneous microbial load, and here the difference is huge because this is a logarithmic scale. So we are in problems. If we look a bit under the scope, then this is the model prediction, what I showed, and this is what the model should predict, a delayed response. It is not possible that the model does this because there is no mechanism. The model instantaneously reacts to a change in temperature. Oh, I should again say no. Okay. What can we do about this? Three things, three methods. We could adapt the model structure, and I will not go into the details for this presentation. Why not? Because, well, this will become too complicated, and it simply says your model is not good enough. Secondly, people in industry, they want to continue using that very simple model. So let's try some other methods, for instance, constraint input optimization. This is at the level of optimal experimental design, or what we call advanced processing of static, very easy to obtain data, and this is at the level of the experimental implementation itself. So let's continue with the second, method number 2. And what we say is that what we allow is a jump from a temperature T1 that can take any value in between T low and T high, a jump off at maximum delta T max, up or down, whatever. Okay? To avoid microbial lag, and you can simply check it in the literature, biologically speaking, the change in temperature should be limited to 5 degrees C. Microorganism can adapt to an instantaneous change of 5 degrees C, not more. Of course, this is at the expense of the information content in the experiment, as we will see. So T1 is end of the optimization T1 coincides with the lower limit, and T2 is just T1 plus delta T max, so plus 5 degrees C. So we do an experiment of, say, from 20 to 25, wherever you position T low. Which is of course not very interesting because the information content is quite low. That's why we continued with, well, let's induce 2 steps. If you induce, or if you allow 2 steps with the delta T max in the vertical direction, immediately you need to impose a second constraint, namely the delta time shift. Otherwise, the algorithm itself will immediately converge to, well, let's make this one big step of 2 times delta T. As we had in the beginning, it was one big step. Why is this delta T shift needed? The delta T shift is also biologically related parameter. It's related to the generation time of the microorganisms. And you need to stay at the same temperature for at least 2 generation times. Then you're sure that the population is adapted to the new environment, to the new temperature. And again, to avoid this intermediate lag, our delta T must be smaller than 5 degrees C. So we have a constraint in this direction, the time direction, and we have a constraint in the vertical direction. Optimization of this type of profile, starting from an initial guess. This is our optimized profile with 3 steps, and the minimum length here is delta T shift. And here is what happens, and this is our prediction of the model. So this is all computer-based, simulation-based. Information content, or condition number 43 almost. In practice, we are quite happy with this result. You might appreciate the quality of the experimental data collected for this experiment. This is exactly the same profile now applied to a bioreactor, and then collecting the data, and you see what happens as a response to this shift in temperature, this profile. This is the simulated profile through the data. Quality, estimation of B and T minimum, contour plot looks like this. Okay. Now to check whether we have convergence, we do a second experiment now on the basis of our last estimate of the parameters, so the P1. We redesign on the computer an optimal experiment. This is what we compute. We apply it in practice. This is the second experiment. Again, and I did not the experiment myself, so I have also a lot of appreciation myself for the quality of this experiment. Almost perfect fit model experimental data. Joint confidence regions, and you see now that the 2, P1 and P0, are within the same 95% confidence limit. So we have indeed convergence. What we last but not least did is a global identification of the 2 experiments that we collected. Okay? And you see that for this third experiment, it's always made nominal with respect to the parameter set that we are after. You see that the experiment 1, experiment 2, and the estimate on the basis of 2 experiments, they're all in the same confidence interval. So we have indeed obtained convergence. Okay, so conclusion, we need small steps, but this is not okay. We need a series of small steps. There is a trade-off between information content and model validity, and this is not obvious from the model itself. It's imposed, it's induced by the biological reality, by the system itself. If you simply do it not knowing anything about microorganisms, this will not work in practice. And secondly, I demonstrated or I illustrated that the sequential experimental design procedure, it seemed to work if we start sufficiently close of the optimal values. Alternative advanced processing of static data. You will remember that the true optimal situation was the following, that we have an experiment in 2 steps. A first phase, if I remember well, at 15 degrees C, then we have a temperature jump, and then we continue at 35 degrees C. correct? This is what we had 10 minutes ago. So that was our true optimal experiment. Well, can we find back or can we reconstruct that experiment without running into the problem of model validity, because we induce an intermediate lag because of that huge jump of 20 degrees C, 15 to 35? Well, we did the following. It's relatively simple. We do a static experiment at the temperature 15 degrees C, and we make it very accurate. The data collection is a very high sampling rate in the beginning. Observe that this is time in hours. Very highly sampled in the beginning. At 35 degrees C, of course, the organisms grow a bit faster, a lot faster. So this is again time in hours, and we continue here. This is our experiment, static experiment at 35 degrees C. And now we will compose ourselves the optimal experiment, part of the data taken from the 15 degrees C experiment, part of the data taken from the 35 degrees C experiment. In the following way, the only degree of freedom, as I said, was the time, T shift. Once we know T shift and we have optimized it before, then we can project this level population density to the curve of 35 degrees C, by which we have selected the two relevant data windows, the first part of the experiment at 15 degrees C, the second part at 35 degrees C. Okay? If we now concatenate in a way these two windows, we artificially, and I stress it's fully artificially, the blue dots and the green dots, we artificially constructed a response to a real step. It's artificial, yes, but it still reflects the ideal situation that the model instantaneously reacts to a jump of 20 degrees C. Now you might say, why do you do this? Because this is not included in the model, your model is not valid. Well, the point is that in practice you will very rarely encounter a temperature jump of 20 or 25 degrees C. In practice, temperature is increasing slowly, linearly, or a change in temperature of a few degrees. Still, we are after the two parameters B and T minimum. And if on the basis of these data we can find them, say, with two experiments, we're very happy with that. Although this is not a realistic response as it could occur in practice. So these are the estimates. I will not go into the details, numerical values. You will find them later in the slides. But again, it is the same quality of estimation if you would compare the joint confidence region. So by artificially composing the experiment by two windows, one at the lower temperature and one at the higher temperature, we circumvented the problem of the model validity. That was a way out. Okay, let's now move on to, this was two parameters, B and T minimum, the linearly, or for you, the linearly increasing part of the maximum specific growth rate. Here it is again. No. A four parameter problem. Again, this is our lag phase, exponential phase, stationary phase. The three phases in our kinetics: lag phase, maximum specific growth rate, the log linear part, and the asymptotic part. This is modeled here. We do experiments at many different temperatures, but now over the full range, so not only the linear part of the kinetics. And what we obtain is the following set of data. This is mu max, not the square root, but mu max itself as a function of temperature. What you see here is a collection of over 100 growth curves collected in our lab. So that it's a big piece of work. It's the collection of, I guess, 3 PhDs in the past of data curves. Okay, you easily recognize the standard form of the kinetics, and in current practice in food processing, we are more and more interested in what happens here. This very abrupt and very fast decrease of the specific growth rate at the maximum temperature, Tmax, which is somewhere here, around 45, 46 degrees C. There is a mathematical model proposed already in '93, and it contains 4 parameters. I show them here. There's again the minimum temperature for growth. There is an optimum temperature for growth. This is the same as in the other model, the easy model. There's now a maximum temperature for growth, and there is a maximum, an optimal value for the maximum specific growth rate as a function of temperature. Now, the natural extension of the previous problem is find optimal estimates not for characterizing this part of the curve, but for characterizing the full part of the curve. And you immediately see the problem. The problem is to collect data in this temperature window, say in between, let me see it from a bit closer, in between 40 and 46, where the temperature, where the specific growth rate drastically reduces from its maximum value to almost 0. Performing an experiment here is extremely difficult. Reproducibility is by no means guaranteed, and this is also what we see in our experimental data. So if we do experiments, we should stay away from this maximum value, although we want to identify it. Similarly, we want to stay away from the minimum temperature, simply because the experiments will last too long. Way too long, weeks, months. So there is a window where we want to work, still wanting to find optimal values of these 4 parameters. To give you an idea of the validity of this model, here you see a linearly increasing temperature profile. This is a validation result, population density as function of time. Here you see a decreasing profile and then remaining constant. This is the population density as function of time. So this model, it works. Finding the parameters is something different. Quality depends, of course, on the model structure, as we said, and find now the 4 parameters. Static data collection. Here you see a detail of the previous plot. This is all what we have available in the lab on this microorganism E. coli K12. About 120, if I'm not mistaken, growth curves. So it's not the idea, if you want to characterize your single microorganisms, to spend 3 PhDs on finding these 4 parameters. That's not the idea. We want to do it a bit faster. Still, these are the 4 parameters: T minimum, maximum, optimum, and the mu opt. Dynamic data collection, where we vary the temperature profile as function of time. We want to be more efficient with respect, of course, to time and labor. And we will design these experiments, of course, with the technique of optimal experimental design for parameter estimation. Okay, I simply repeat the setting of the problem. So the output is again the natural logarithm of our population density. In this case, a parameter vector, it contains 4 parameters, 4 unknowns. So this is a bit more complicated now. Weighting matrix, model output sensitivity matrix, again the same problem. In this case, we decided to work with the D criterion. Okay, just for a change, but there were other reasons, mathematical reasons as well, but this would lead us too far. So we will maximize the determinant of the Fisher information matrix, just to obtain the maximum global accuracy of the parameters. This n is a population density for which we need a partial derivative with respect to the parameters. We have the state equation, the ordinary differential equation describing population density as function of time and temperature via mu max. And for mu max, we have this CTMI cardinal temperature model with inflection point. So this model containing the 4 parameters, which we then need partial derivatives, and so on and so on. Model validity, we have learned from the past, so we will not allow abrupt temperature changes. We introduce it from the beginning as an additional constraint in the experimental setup when we do the optimization. And we will not allow that, well, Umax will change, but the temperature changes themselves will be limited, the amplitude will be limited, the gradient. So this is the set of nominal values that we extracted from all of our static experiments available. The values are not that important, but it gives you some idea. If later on during a reception someone asks you what is the optimal temperature of an organism, if you say 40 degrees C, I don't know in Fahrenheit, but anyway, 40 degrees C, 90% is very close. That's a good rule of thumb for most organisms, especially the pathogenic ones. Optimization of what temperature inputs. Again, we made it a bit simple, not because we do not want to do infinite dimensional optimization, but we need to implement these experiments in practice, in a bioreactor or in a water bath or whatever situation. We need to implement it. So we have a lower temperature, an increase or decrease with some gradient, and a second temperature, a final temperature level. Lower temperature limit, superimposed, higher than the minimum temperature, and a maximum temperature smaller than T max, for the reasons I explained before. And for reasons of model validity, the gradient is limited to 5 degrees C per hour. And we tested it on many different organisms and it works. This is pretty much okay. We tested a number of situations. Remember, we have to identify 4 parameters, so we tried simultaneous optimal experimental design. We tried what we call sequential, and I have to agree that, well, we invent all of these abbreviations ourselves, so no one understands them. Anyway, simultaneous experimental design, sequential, and then global. It will become clear in a minute, I hope. Simultaneous, easy. We are allowed to do 1 experiment. On the basis of this, we need to find the 4 experiments, the 4 parameters. Okay, 1 experiment, 4 parameters. So the dimension of the parameter vector is 4. Of course, computationally speaking, this becomes quite complicated if you work it out in practice. And it's very questionable that you can identify from a single experiment the 4 parameters that fully characterize the maximum specific growth rate as function of temperature. We tried, and I will show you the results in a few minutes. Sequential, this is 2 by 2 estimation. We fix 2 and we estimate the other 2. We can do it by 6 experiments. Well, with 4 parameters, all of us, we know that there are 6 combinations possible. And we do an intermediate, and that's crucial here, we do an intermediate update of the nominal values and the parameter estimates. By which I mean that if in the first run we start with nominal values for the 4 parameters and we estimate P1 and P2, we design the experiment, then in the second run, say we then identify P1 and P3, then we will use the value that came out of run 1 for P1 and the value for P2 that also came out of run 1. You see? And this continues. So once an updated value becomes available, we use it. Of course, this is arbitrary in a way because there are many sequences possible in this design, which, as we saw it in practice, does not lead necessarily to convergence. Global identification, by which we understand this is also 2 by 2, but here we keep in the 6 experiments that we perform, and this is already written out in terms of the maximum temperature for growth, mu opt, the minimum, and I'm forgetting one, the optimal one, yes, it's here. We still keep the original known nominal values. We do not do intermediate updates. We do the designs independently. And then in the end, we globally identify the experiments. I keep saying no. Do you want to proceed? No. But I keep proceeding, if you don't mind. In simulation, what does it say? 3 steps. The optimal criterion. We simulate our curves. Let me go immediately to the results. We have tested it for 3 different true parameter sets. The simultaneous and the global, it more or less works. The sequential, we tested many different sequences of P combinations, as I explained. There is no one single route if you want to do this and do the intermediate update. So we did a Monte Carlo analysis and tested many possible different sequences here. Now, the important conclusion is that if these are our nominal values, so the starting values, and these are the true values, we have 3 sets. This is set 1, this is set 2, this is set number 3. We see here the simultaneous results all 4 parameters together, 1 experiment. Sequential, 6 experiments but intermediate updates. Global, 6 experiments, no intermediate updates. Global identification on 6 experiments. And what we see is, strangely enough, that in simulation, this is because of the noise level that we can control, that simultaneous and global, they more or less coincide, given the accuracy. Sequential, and we picked a bad example, it's out of the line. Sequential, it does not work, especially not for these biological systems. I'm not saying in general, but for the systems at hand, it doesn't work. So that's why we continued with simultaneous and global, now in practice on experimental data collected in bioreactors. Again, our bioreactor, the same microorganism, cell density was measured by plate counting, so colony forming units, all the same. Here is our bioreactor. These are the repetition of what we have already available, extracted from this nominal parameter values from all of our static experiments, and we imposed as an additional constraint in the design a maximum gradient for the change in temperature as function of time. Final time of the experiment was fixed to 38 hours, so that we could run, including preparation of the experiment and processing of the experiment, that we could run everything in 1 week. 5 working days. Preparation is also 2 days, 2 days and a half. Global identification, what you see here is the 6 experiments designed and then performed in practice. Again, appreciate the quality of the experimental data as they are collected here. So these are the 6 different experiments, each for the 2 parameters that we wanted to identify, the other 2 kept at their nominal value. And what you see here is global identification for the 4 parameters. It works, at least at first sight. Simultaneous, and I go immediately to the result, to the comparison. For this organism, the comparison, global identification, or the 4 parameters all together in 1 experiment, you can hardly distinguish, biologically speaking, what is the best value. Why? Because here we, in our selection of nominal values, hidden in this is a set of 120 static experiments. And that of course explains why the difference between the 2 methods is so small. What we see here, and this is the transfer of what we have been doing to another organism, it's a yeast responsible for spoilage of mayonnaise, ketchup and these things. A very common yeast, we don't like it. There is only 1 literature reference, and these are the nominal reported values. When we did preliminary static experiments, this was the best we could obtain in the lab. A very difficult organism to work with. It's not a bacteria, it's a yeast. Nominal values that we selected, also the range that we selected, also the same gradient that we did allow. Global identification of 6 optimized experiments, 2 parameters optimized, the other 2 fixed to the nominal values. Simultaneous, well, these correspond more or less to what is reported in the literature, as I indicated before. Simultaneous, so 1 experiment that we could do, and you see immediately that we are out of line with what is reported, especially the nominal value for T minimum. And also, which is an important value, the maximum temperature for growth. Here we predict 38.3, which is, well, that's a bit too far away from the reality. This does not work because of the quality of the original nominal material. If I go back one slide, this is much better. Global identification, experimental burden, only 6 experiments. But you need to design them very carefully and perform them even more carefully than the design. Conclusions: 3 optimization strategies, simulation study, sequential design. Do not trust it necessarily. It could work, but we don't know what is the optimal sequence. If we go for the global estimation with 6 experiments, we can do the job. 6 experiments to identify 4 parameters is not too bad after all. And secondly, for simultaneous, so one single experiment, the ultimate dream, we do one experiment and we extract all unknown parameters. This is basically not possible. I will skip the case study number 2, if that is fine with you, but I will come immediately to the conclusions, but you will find them in the handout, or the slides are published as well. Okay. In that second case study, we made a trade-off between optimal identification and optimal process performance for a bioreactor. And I simply concentrate 1 second on this slide. So this is optimal production in the bioreactor. And this is the condition number of the Fisher information matrix characterizing identification quality. And what you see here is that the performance of the bioreactor, when we do simultaneous identification of our parameters, it remains pretty high. It only decreases quite rapidly when we are very close to an optimal condition number. But for the majority of cases, you can find a very good trade-off between maximum production, maximum productivity, so your process is running in full operation and still being able to identify the parameters. This is also being tested in practice now. So then I come to my conclusions. Combination of other criteria. We have D criterion, we have the E criterion, modified E criterion. We are considering curvatures as introduced by the speaker this morning as well. Combination of criteria in a multi-objective setting. Combining model discrimination and parameter estimation. The issue was raised this morning as well in the conclusions. And finally, can other criteria be combined to cope with each other's drawbacks? One criterion is good for this one, accuracy, decorrelation, and so on. Can you combine it, or do you need to do it in an iterative way? Issues where I believe that people working more on the theoretical side might want to contribute. My last slide, these are 3 websites, and again you will find them later on. These are 3 platforms. The one is concentrating on the chemical industry. The second one is concentrating on wastewater treatment, biological wastewater treatment. The third one is concentrating on predictive modeling in food products. 3 platforms open for collaboration with industrial partners that we are now actively involved in, and considered as spin-offs of our academic research in the Center of Excellence and in our own research division. Thank you for your attention. If you have any questions. And I'm close to, well, 1 minute left.
Yeah, we have time for a quick question.
So there's a lot of stuff, a lot of work on compartmental models and these kind of models where differential equations with polynomial coefficients. And people do things like look at the steady state solutions algebraically. And may lead to a different, well, one of the benefits of doing that is they may lead to a different parameterization. For example, if you set a little differential equals 0 and solve, you get some natural parameters which are identifiable in steady state, things like that.
Yep.
Have you thought of a reparameterization by looking at the algebraic analysis of equations?
We did. In a way, it corresponds to, in control theory, singular perturbation, and if you find, this is, I guess, what you're pointing at, or similar methods. The point is that, in general, yes. For chemical systems, yes. For biochemical systems, most often no, because the steady state response does not necessarily reflect dynamic behavior of a microorganism, and that's the main reason. And the point is not that the techniques are not there, the point is that the models, the quality of the models is not good enough at this moment. Reflecting that the model should be able to represent the microorganism under both static and dynamic conditions. These models hardly exist these days. They are improving. But if you then go to the other spectrum in the area of systems biology that we're also active in, yeah, this is at this moment so tremendously involved and complicated.
The algebraists go around using words like stoichiometric.
Exactly, and if you then go a little bit deeper inside, the mu is modeled by a pseudo-steady state assumption. Stoichiometry times flux factor equals 0. Because we need to solve that system of...
It's not relevant here because it's a different side of the problem.
yeah, correct.
Another quick
Don't
question.
you think even if you were able to reparameterize, because you're using criteria such as
Could you speak a
d-optimality,
bit louder?
even if you were able to reparameterize, because you're using such criteria as d-optimality, you would have encountered lack of invariance with respect to reparameterization.
Yeah, correct.
Your science. So
Yeah, I agree.
that's actually criticism for d-optimality. Oh, okay. Some more questions.
Okay, last one.
Oh, well, we've got half an hour. Really?
Well, I'm here for coffee, you know. I'm still here for the coffee.
Oh no, I was going to have tea actually. You put up the results of a simulation experiment in which you had compared 3 different ways of doing the experiment.
Yeah.
Did you do any sensitivity analysis to this? I wonder if your results depend on whether your guesses or initial guesses are good ones or not. If you had horrible initial guesses, might the simultaneous one be better?
You might have missed it, or maybe I was too fast because
I might
was
have missed
running.
it.
No, it's my fault. Whatever.
I insist, yes.
But we tested 3 initial or 3 true parameter sets that were quite far away from each other. So that's even more than we had the initial estimate, nominal values, and then the true parameter set, we picked 3 different true parameters.
So the nominal and the...
Yeah.
Okay, as
And
well
we
as...
did it for the 3.
Yes, as well as doing 3 different kinds of
Yeah.
experiments. Okay, yes, I did miss that. Thank you, student.
Okay, well, thank you very much indeed. So we have coffee coming up, so there'll be plenty of extra time to carry on. Unless you particularly want to do this in the group session, you can ask if you